There is a place on this planet where sunlight has never reached, where pressure exceeds 1,000 times what exists at the surface, where temperatures hover just above freezing, and the darkness is not simply an absence of light. It is a physical condition, total and permanent. It sits at the bottom of the Mariana Trench in the western Pacific Ocean, so deep that if Mount Everest, all 8,849 meters of it, were lowered into this abyss, its summit would still disappear beneath the waves by more than 2 kilometers. For most of human history, this place was assumed to be dead, barren, a pressurized void incompatible with any known form of life. Scientists called it the azoic zone. The logic seemed sound. No light, no warmth, no photosynthesis. They were wrong. What has been discovered at Challenger Deep, the deepest measured point on Earth, at approximately 10,935 meters, is not a void. It is an ecosystem, a geological archive, a chemical laboratory operating on timescales that dwarf human civilization. And buried within its sediment layers, scientists are uncovering answers that reach far beyond this trench, toward the most fundamental questions about life itself. How did organisms not only survive here but evolve to thrive under conditions that would kill nearly everything else on the planet? What is preserved in sediment layers accumulating for tens of millions of years? And what does this place, visited less often than the surface of the moon, tell us about where else in the universe life might exist? The investigation begins here. The Mariana Trench sits approximately 200 kilometers east of the Mariana Islands, a crescent-shaped canyon stretching 2,550 kilometers in length. Its formation is tectonic, the ancient Pacific Plate, dense and cold after nearly 180 million years of cooling, subducts beneath the Mariana Plate at a steep angle, carving a boundary trench of extraordinary depth. At its southern end lies Challenger Deep, measured by modern multi-beam sonar and submersible pressure instruments at approximately 10,935 meters. The variance between surveys is minor. What remains consistent is the scale. More than 10 kilometers of vertical descent through absolute darkness. The trench is not geologically dormant. The subducting Pacific Plate generates ongoing seismic stress along its boundary, releasing periodically insignificant earthquakes. A 7.7 magnitude event struck the region in 2016. This is a structure that is still, in the slow logic of geological time, being formed. Measuring its depth with precision requires layering multiple sonar passes with direct pressure measurements from submersibles, accounting for variations in water temperature and salinity that affect acoustic velocity. The accepted figures are the result of decades of iterative refinement. What they confirm is a location so extreme it belongs, in almost every measurable parameter, to a different world than the one above it. At Challenger Deep, pressure reaches approximately 1,086 atmospheres, equivalent to 110 megapascals, or roughly 16,000 pounds of force per square inch. A foam cup lowered to this depth returns the size of a thimble. An unmodified human body would not be crushed gradually. The gas-filled cavities, lungs, sinuses, intestinal tract, would collapse instantaneously. And yet life is abundant here. The mechanism lies at the molecular level. Proteins, the functional machinery of all living cells, fold into three-dimensional structures that allow them to perform specific biochemical tasks. Under extreme pressure, this folding collapses. Proteins denature, losing function. To counteract this, deep trench organisms accumulate a compound called trimethylamine oxide, TMAO, in their tissues. TMAO stabilizes protein structure under pressure by altering how water molecules interact with protein surfaces at the molecular scale. In hadal species, TMAO concentration scales almost linearly with depth. It is a finely calibrated biochemical solution, refined through millions of years of evolutionary pressure. Temperature at Challenger Deep sits between 1 and 4 degrees Celsius. This cold slows enzyme activity, the metabolic reaction rates that sustain cellular function, requiring modified enzyme architectures with greater molecular flexibility at low temperatures. Light is entirely absent below 200 meters. The entire energy budget of the hadal ecosystem depends not on solar radiation but on marine snow, particles of organic matter, fecal material, and dead organisms that drift downward from the sunlit surface ocean, carrying accumulated chemical energy across more than 10 kilometers of water column to the trench floor below. The hadal zone, the depth stratum below 6,000 meters, covers less than 0.2% of the ocean floor. What it contains defies its size. Hadal snailfish, Pseudoliparus swirii, have been recorded at depths exceeding 8,000 meters, the deepest confirmed fish sightings in scientific history. Their bodies are translucent, gelatinous, and lacking rigid skeletal structures or gas-filled swim bladders, both of which would be destroyed at hadal pressures. Their tissue TMAO concentrations are among the highest measured in any vertebrate. Despite this, footage from deployed landers shows them moving with speed and agility, competing actively around food sources in behavior that mirrors surface fish feeding. Below the fish, amphipods dominate. The species Hirondelia gigas occurs at Challenger Deep in densities that surprise researchers given the apparent scarcity of food. Their digestive systems contain enzymes capable of breaking down wood cellulose, a capacity that suggests evolutionary exposure to terrestrial woody debris transported by deep ocean currents from continental sources thousands of kilometers away. Single-celled foraminifera carpet the sediment in enormous concentrations, their mineralized shells recording chemical signatures of the ocean conditions in which they formed. Among them, xenophyophores, the largest individual cells on Earth, some exceeding 20 centimeters, extend their branching structures across the trench floor. At the base of the food web, microbial communities drive the decomposition of all incoming organic material, cycling nutrients back into the water column. Some of these bacteria are obligate piezophils, organisms that require high pressure to function and cannot survive at atmospheric conditions. Beneath the organisms, the trench floor accumulates sediment at approximately 1 to 2 millimeters per thousand years. This rate is imperceptibly slow in human terms. Over tens of millions of years, it produces deep, largely undisturbed sediment columns that preserve a continuous record of Earth's oceanic and climatic history. This is what makes hadal sediment uniquely valuable. Unlike most ocean floors, where currents redistribute particles, organisms disturb layering, and geological events interrupt the sequence, deep trench sediment accumulates with minimal disruption. Each centimeter of depth corresponds to a measurable period of geological time, sealed and intact. The material arriving in these layers carries layered records. Oxygen isotope ratios in foraminiferal shells serve as proxy climate thermometers, reconstructing ocean temperatures from before any human record. Wind-blown continental dust carries signatures of ancient atmospheric circulation. Cosmic micrometeorites mark the passage of geological time. Most striking is the discovery of metabolically active bacteria recovered from sediment layers estimated to be millions of years old. These organisms are not dormant. They are functioning, conducting cellular maintenance at energy levels so low they approach the theoretical minimum for biological activity. They persist because the chemical gradients sustaining them have remained stable, undisturbed, across timescales that encompass the rise and fall of entire geological epochs. The sediment at Challenger Deep is, in the most literal sense, a library. Its contents span tens of millions of years of planetary history, written in chemistry, biology, and physics, and largely unread. In 2017, a research team from Newcastle University published findings from amphipod crustaceans collected at Challenger Deep and the Mariana Trench. The lipid tissue of these organisms, the biological fraction where persistent compounds accumulate, contained polychlorinated biphenyls, PCBs, at concentrations exceeding those found in crustaceans from heavily industrialized coastal river systems. They also contained polybrominated diphenyl ethers, PBDEs, flame retardants manufactured almost exclusively in the 20th century. The pathway is not mysterious. Persistent organic pollutants attach to organic particles in the surface ocean. As those particles sink, as marine snow, the chemicals descend with them. A trench geometry, where material falls in from an enormous surrounding catchment area and accumulates in an enclosed basin, concentrates these compounds over time. The trench functions as an inadvertent chemical trap. In 2019, microplastic fibers and fragments were confirmed in sediment samples from Challenger Deep. These particles are now physically embedded in the geological record, a datable stratigraphic horizon marking the arrival of industrial civilization's output at the deepest point on Earth. The organisms here did not evolve in a chemically contaminated environment. The biological consequences of this contamination for hadal ecosystems remain almost entirely uncharacterized. The baseline data required to measure change does not exist because we began contaminating Challenger Deep before we had finished understanding what lived there. Fewer than 20 people have physically reached the floor of Challenger Deep. More than 600 have traveled to space. The first descent occurred on January 23, 1960. Swiss oceanographer Jacques Picard and U.S. Navy Lieutenant Don Walsh descended aboard the bathyscaphe Trieste, a steel-sphered personnel capsule suspended beneath a float filled with aviation gasoline. The descent took 5 hours. They spent 20 minutes on the bottom, at a recorded depth of 10,916 meters, before ascending over 3 hours. No crewed vehicle returned for 52 years. In March 2012, James Cameron descended alone in the Deep-Sea Challenger, reaching 10,908 meters and spending 3 hours conducting scientific observations before a hydraulic fault prompted ascent. In 2019, Victor Vescovo descended in the DSV limiting factor, reaching 10,928 meters, the deepest confirmed crewed dive in history, and conducted multiple dives over the course of a single expedition, producing the most detailed sonar mapping of Challenger Deep yet achieved. The engineering demands are extreme. Every component must survive pressures exceeding 1,000 atmospheres through both descent and ascent. Modern deep vehicles employ thick-walled borosilicate glass viewports. Glass becomes stronger under compressive loading, making it superior to metal for pressure housings, and titanium alloy structures selected for strength-to-weight ratio and corrosion resistance under deep ocean conditions. Each crewed and uncrewed visit has returned findings that contradicted established expectations. The pattern is consistent. At maximum depth, the unexpected remains the norm. Challenger Deep has answered many questions about the physical limits of life on Earth. Its most consequential contribution, however, may be to a question that extends far beyond this planet. For most of astrobiology's history, the search for potentially habitable environments beyond Earth operated on assumptions derived from the surface biosphere: liquid water, moderate temperatures, solar energy, atmospheric protection. Applied across the solar system, these criteria produced a short list of candidates. Challenger Deep rewrites the criteria. Life at maximum depth requires no sunlight, no moderate temperature, no atmospheric pressure. It requires only liquid water and a chemical energy gradient, and these two conditions are met across environments distributed far more widely in the solar system than any planetary surface. Europa, the fourth largest moon of Jupiter, harbors a global liquid ocean beneath an ice shell, estimated 60 to 150 kilometers deep, maintained by tidal heating from Jupiter's gravitational influence. Its ocean floor is theorized to host hydrothermal activity, the same chemical energy source that supports entire independent ecosystems on Earth's deep ocean floor. Enceladus, a moon of Saturn, actively vents water vapor plumes from its southern pole, carrying molecular hydrogen, a chemical fingerprint of hydrothermal reactions, and complex organic molecules. Liquid water, heat, and organic chemistry have all been confirmed from orbit. Neither moon has been confirmed to host life. But the discovery that life thrives at Challenger Deep, in darkness, under crushing pressure, near freezing, sustained entirely by chemical gradients, transforms both locations from speculative curiosities into scientifically serious candidate environments. What Challenger Deep ultimately demonstrates is this: life is not a fragile anomaly requiring a narrow set of improbable surface conditions. It is a robust chemical process that has colonized the most extreme environments this planet offers, persisting across geological time, adapting to conditions that nothing in its original discovery prepared science to anticipate. If it exists here, at the most inhospitable point on Earth, sustained by the slow chemistry of falling particles and ancient sediment, the question is no longer whether similar conditions exist elsewhere in the universe. The question is whether we are looking hard enough. Our world is full of incredible stories waiting to be discovered. Let us know if you enjoyed this journey, and be sure to like, subscribe, and join us again for more fascinating documentaries.